in the middle of the week. It's a bank holiday. Is it okay if we record this transmission?
In principle, yes.
Yes.
But in the end, I'd like to show some unpublished data yet. So
right. So what we can do is we can stop when you're going to reach the point, if you prefer that. Yeah. So William, please take care of this, okay? William, ¿me estás escuchando? Para cuando llegue ese momento, paramos la transmisión.
Listo, profe.
Donde él dice que vamos a mostrar algunos resultados que no han sido publicados.
Okay.
¿Vale? Okay, Richard, so I think we can start, yes? I'm going to...
So I guess I should share the full screen, right?
Yes, so I'm going to make an introduction first before we start. So, buenos días a todos y todas. Es un gran placer y un honor tener como hoy a nuestro invitado, colega y amigo, profesor Richard Hillner, de la Universidad de Groningen, del Instituto Cernique para Materiales Avanzados. El profesor Hillner es profesor desde el 2018 en el Instituto Cernique, en la Universidad de Groningen. Previo a eso, entre el 2011 y el 2018, fue el líder de un grupo de investigación en la Universidad de Bayreuth, en la sección de Experimental Physics 4, en el área de propiedades electrónicas y ópticas de macro y de nanoestructuras macro y supramoleculares en el estudio de las propiedades funcionales y biológicas en la Universidad de Bayreuth. Fue donde él mismo se graduó de doctor. Entre el 2008 y el 2011 fue un investigador postdoctoral en el Instituto de Ciencias Fotónicas de Barcelona, en España. Y entre el 2003 y el 2007 fue un asistente de investigación en la Universidad de Bayreuth, en el área de espectroscopía óptica de polímeros conjugados individuales. A nivel de su educación, el profesor recibió su habilitación en física experimental en la Universidad de Bayreuth en el año 2017 con su tesis Transporte de energía en agregados supramoleculares naturales y sintéticos. En el 2008 se recibió de doctor en la Universidad de Bayreuth. Su diploma, es decir, lo que es pregrado para nosotros, en el 2003. Y entre el 1996 y 2003 hizo estudios de física entre la Universidad de Bayreuth y la Universidad de Exeter en el Reino Unido. El profesor Richard tiene varios premios. El más reciente, en el 2019, fue el premio al Teacher of the Year en el área de nanociencias en el programa Top Master y la nominación al Teacher of the Year en la facultad de la Universidad de Groningen. En 2014 obtuvo el premio de ciencias en la Universidad de Bayreuth. 2013 el premio Michael Sturge por investigadores jóvenes en el campo de espectroscopía de materia condensada, y tiene mención de summa cum laude en su tesis doctoral. Richard, it is a pleasure to have you with us, and please, you can go ahead with your presentation.
Yeah, thank you very much for this kind introduction, and of course for this invitation. It's always a pleasure to be a guest in Cali. Today in this colloquium, I want to show you a little bit recent work that we did related to supramolecular nanostructures, and specifically looking at energy transport and control of energy transport in supramolecular nanostructures and even larger superstructures, and specifically how we can make use of properties of excited states and exciton landscapes to control energy transport. So I think I don't need to introduce why the energy transport is important. Just as an example, in organic solar cells for sustainable energy conversion, so here on the right side, we have a few examples. So in the bilayer architecture, so you have two materials, a donor material, usually conjugated polymer or a large organic molecule. And at the bottom, the blue one here is an acceptor material, which is electron accepting So at the interface, you get a charge separation to convert energy, light energy after absorption, into a current. Or if you talk about these bulk heterojunction architectures here, where you just mix both materials, and here I zoom in on the right side. So this yellow spaghetti type stuff here represents a polymer, a conjugated polymer, which absorbs light to create an excited state somewhere here in the center. And this energy has then to transfer to this interface to the accepting material, in this case, a C60, C70 fullerene, which is a very strong electron acceptor. And then the charges are extracted out to the direction through the fullerenes and the polymer towards the electrodes to get a current. And on the other side, this energy transport is also a highly important process in photosynthesis, which, as you probably know, is well, the basis of all life on Earth. And as an example, I want to show the photosynthetic apparatus of purple bacterium. That's a bacterium that lives at the bottom of lakes and ponds and harvests sunlight. And this photosynthetic apparatus, here's on the top left, here we have an image of a single cell bacterium, and the photosynthetic apparatus is out here in the cell membrane. And the zoom into this photosynthetic apparatus is shown here as a sketch. So you have these ring-like structures, so smaller rings out here surrounding a larger ring in the center, and in the void of the bigger ring in the center, you have a so-called reaction center. And what is in fact happening in this system is that these rings, they constitute so-called pigment-protein complexes. So you have an organic molecule that absorbs sunlight coming in somewhere. You create excitation energy, and this energy is then transferred within these rings, between rings, and into the reaction center. And in the reaction center, charge separation takes place, which provides some chemical energy for the organism to survive. And if we take a closer look to these energy transfer properties that we have in these photosynthetic apparatus, so we look here at a small section of one of these rings, so-called light harvesting complexes or pigment protein complexes. And we have here in red, we have bacterial chlorophyll A molecules oriented with a phase in the plane of the screen now. And then here we have some aliphatic side chains. And there you have a series of red, marked red bacterial chlorophyll A molecules. And at the bottom here, the face of the bacterial chlorophyll is perpendicular to the screen plane. And so you have a second ring of these bacterial chlorophyll molecules. And they are all kept in this ring-like shape. So you have in total 27 bacterial chlorophylls in each ring. They're kept in this shape and in position by non-covalent interaction between different bacterial chlorophylls and by non-covalent interactions to the protein scaffold. And here just 3 alpha helices for illustration that keep them together. So you have some non-covalent interaction between these pigments, the bacterial chlorophyll A, to protein residues in these alpha helices. So there is essentially no covalent bond involved between molecules and between pigments and proteins. So it's a fully supramolecular architecture here. And this keeps the molecules in a very dense, so as I said, we're talking here about 27 molecules in a ring of 4 nanometer diameter, and at the same time, a very precise arrangement of pigment molecules. And that enables energy transport within this antenna system. So the full distance from where light is absorbed to the reaction center, this can be up to 100 nanometer. So it's a fantastic distance. If some of you might be familiar with organic solar cells, then you're lucky if you have 10 nanometer transport distance. So within 10 nanometers, you have to find an interface to an electron acceptor. At the same time, in photosynthesis, the quantum efficiency is near 100%. That means every absorbed photon here at this position is converted into a charge-separated state in the reaction center. So that part is extremely efficient. You do not waste any photon or any energy quantum. And the whole thing is also very fast. So within sub-picoseconds, few 10 picoseconds, the whole process from absorption to charge separation is done. So nature tells us essentially that if you look very carefully in how you arrange molecules spatially, how you can get long-range energy transport. And one important factor, this is what I will show you at the end of this talk, is that in fact in this full photosynthetic apparatus here on the top left, the energy always goes downhill. The excitation energy always goes downhill in the excited state energy landscape. So there is an energy funnel from these peripheral light harvesting complexes into the reaction center, which drives this efficient and long-range transport. And so these are the aspects that we want to address in my group. So developing systems, artificial systems that are suited for long-range transport, where we can control transport and where we can, well, find ways even to design this energy gradient or this energy funnel within a supramolecular system. And I want to highlight that this energy funnel is not made with different molecules. Yeah. So these, all molecules that are involved in absorbing energy, transferring energy, even in the reaction center for charge separation, this is all bacterial chlorophyll A. And nature manages to get this energy funnel. Now, how does that happen? How does nature essentially tune the excited state energy landscape? And then I will make a short excursion into exciton theory. So let's assume we have a molecule, any organic molecule, bacterial chlorophyll A or other molecules that absorb invisible light. And this molecule is just this gray ellipse up here, and this double-headed arrow is the transition dipole moment. Now, if this molecule is isolated, no other molecules, for example, in a dilute solution, we describe this molecule as just a two-level system. We have ground state where light can be absorbed by interaction with the transition dipole moment. We're in the excited state, and then it can be released by emission of a photon. So we get fluorescence out of these molecules. Now, if we bring these molecules very close together, so the transition dipole moments can in fact interact with each other by Coulomb interaction. So they start feeling each other. That has several consequences. So that would be the situation in nature in these light harvesting complexes, where the molecules are very closely packed. So this interaction, first of all, means that the excitation energy is not localized, as in the case of monomers. Without interaction, the energy is just localized on one molecule. With interaction, we delocalize the energy over the entire aggregate. So where the entire, well, structure, supramolecular structure that we have. The second consequence of interaction is that in this simple picture, the excited states of the monomers have all the same energy, they're isoenergetic, but with interaction, this degeneracy is lifted, and we get an exciton band with as many exciton states as we have monomers in our aggregate. And the third consequence is that the oscillator strength is redistributed in that system. And this redistribution of oscillator strength, which is a very important aspect for our work, depends on how the transition dipole moments are oriented with respect to each other in a structure. So in this case of head to tail or inline arrangement, as shown here on top, the oscillator strength is in the bottom exciton state. This is a situation, if you think about oscillating transition dipole moments of each monomer here, and this is a snapshot. of the oscillating transition dipole moment. Then you see in the bottom state, the dipole moments oscillate all in phase. So you have the sequence, well, negative charge density, positive charge density, negative, positive, and so on. Right? So this is an energetically favorable situation. That's why it's the lowest energy state. And at the same time, this is a state that you can drive by light when all dipole moments oscillate in phase. So this is where the oscillator strength is. And this is a highly radiant state, a so-called super radiant state, and lifetime is about 10 to 100 picoseconds in this situation, usually what you see. Just for comparison, a normal molecule, a normal monomer has lifetimes of 1 to 10 nanoseconds, so several orders of magnitude slower. So you really speed up the emission process by this redistribution of oscillator strength. And for completeness, the top state of the exciton band here, this has no oscillator strength. This is the unfavorable situation in the snapshot where the transition dipole moments of neighboring molecules oscillate out of phase. So you have negative, positive, positive, negative, and so on. And this is a state that you cannot generate by light. That's why it does not absorb, and it also cannot emit. And the main point here is that if you have such a system, and this is called a J aggregate with this inline arrangement of transition dipole moments, you have very short lifetimes. That means you don't have a lot of time to do something useful with the excitation energy once you create it by absorption into this bottom exciton state. It will decay very, very rapidly back to the ground state by fluorescence. So you cannot do a lot useful. Sorry. But you can change the situation if you go to a slightly different arrangement of molecules. So in this case, this would be a parallel arrangement of transition dipole moments shown here. So you simply rotate the molecules by 90 degrees. And again, once you switch on electronic Coulomb interaction between transition dipole moments, you will get a delocalized exciton state. You will get a lift of the degeneracy of the monomer energy level. So you get an exciton band as shown here. And now the top state carries oscillator strength. So absorption from the ground state will occur into the top state of this exciton band. And the reason is that in the top state now, this is the energetically unfavorable situation where transition dipole moments oscillate in phase with negative, positive, negative, positive. And this is what you can drive by light. While these H aggregates, on the other hand, they are not emissive, because what you have is you get quick relaxation to the bottom of the exciton band, to the lowest energy state. And this is now a state where transition dipole moments oscillate out of phase. This is nothing that can generate and can be driven by light. So this is a dark state. So in H aggregates, this is called H aggregates, this parallel arrangement of molecules. They have a subradiant lowest energy state, and they have usually a very long excited state lifetime that can be tens of nanoseconds up to microseconds. So if you tune the mutual arrangement of your molecule from this limiting cases inline or parallel arrangement of transition dipole moments, which we refer to as a supramolecular design of our system, you can tune the excited state energy landscape. So that means in our language, which of the exciton states are optically accessible, carry oscillator strengths. That means the bottom state in J aggregate, the top state in H aggregates. And what you also do is at the same time, you engineer the lifetime of the excited state. That means always the lowest exciton state, lowest energy exciton state, where you can tune the competition with energy transport. In H aggregate, long lifetime means you have a lot of time to transport energy across your supramolecular structure, for example, in these light harvesting systems. Now, in our situation, we certainly want H aggregates, because we really want to aim for very long range energy transport, we really want to go in the direction that we have several tens, hundreds, even micrometer energy transport in artificial systems. I should mention that this lifetime engineering is, of course, not our invention. This has been shown nearly 60 years ago by Avakian and Merrifield on Anthracene crystals. The triplet states in Anthracene have very long lifetimes, microseconds, several microseconds, and they have shown that you can transport triplet energy over microns at room temperature. But for singlet excitons that we are talking about, we are still limited by the record of nature, some 100 nanometers energy transport. Now, coming back to our system, so what we want to do is clearly building an H aggregate, and we talked to our favorite chemists. That was still during my time in Bayreuth in Germany. And the chemists came up actually with this very, very large molecule shown here. So the center, the core of the molecule is a so-called carbonyl-bridged triarylamine. So you have these aryls. These are these phenyl rings here. They are bridged with carbonyl groups. That's this part here, and the amine is the nitrogen in the center. So it's an aromatic molecule essentially. And at the edge of these molecules, you have in green labeled amide groups. And attached via a short aliphatic spacer, you have naphthalimide bithiophene. So this naphthalimide is this part, bithiophene is this part down here. If you look at the absorption emission of this molecule, well, the absorption spectrum is here on the left, a maximum around 450 nanometer, and emission strongly Stokes shifted at around 600 nanometer. But it becomes more interesting if you look how these spectra, or from which groups these spectra arise. So what we typically excite would be at 450 nanometer in the absorption maximum. And if you take a close look, the absorption is actually given by the core, the CBT core, so this orange part of the molecule, and the naphthalimide bithiophene, the peripheral group of this molecule. It's really just the sum of the two. But so when we excite at 450 nanometer, we excite both the core and this peripheral naphthalimide bithiophene group. But emission only comes from this peripheral group. So this molecule in itself is an energy transfer system. If we excite the core with light, energy will be transferred very quickly with a high efficiency to the naphthalimide bithiophene, and then will be emitted. And the idea behind this peripheral group, which is a strong emitter, strong orange emitter, is that if you, what I mentioned a few slides ago, if you have an H aggregate, the lowest energy state is a subradiant state. It's very, very weakly emitting. So if you want to observe anything from an H aggregate, you better do something to it that it starts giving light. And this is the job of these emitting peripheral groups. Now, this molecule turned out to be a very, very nice system. So it shows a very robust self-assembly. And here's an AFM image of a dilute solution. So you see these long fibrillar structures here. The length, this is about a bit more than a micrometer. And if you look at the height profile along this green dashed arrow here, that's below, and we're talking about a height of about 3 nanometer. If you compare that with the molecular diameter of our molecule, which is about 6 nanometer, you see that the diameter of this nanofiber in the AFM image is given by the diameter of the molecule, essentially. So we have fibers with molecular diameter that live at very low concentrations. That is 70 nanomolar. This is for those of you who are familiar with self-assembly. This is extremely low concentration for self-assembled systems. Now, how do we envision the structure of our nanofibers. That's shown here in a sketch. So just to remind you, we have in the core of the molecule this orange part here. This is aromatic core, and these aromatic molecules tend to pi stack via attractive van der Waals interactions. So the core of these molecules will tend to lie on top of each other, and this is supported by these hydrogen bonding amide groups, the green groups. So we have the CBT core stacked perfectly on top of each other due to van der Waals interactions, supported by these chains of hydrogen bonding amide groups here. Translated to the introduction I provided you a little bit earlier, so we have all transition dipole moments lying parallel next to each other, so we have a perfect H aggregate in the core of this molecule. And the periphery out here, this is the peripheral groups. This is just a disordered tube of molecules, so the periphery does not do any H aggregation or whatsoever. Yeah, we know that from different measurements. So the only H aggregation that occurs is in the core of these nanofibers. Now, how can we image energy transport? And we did that in these first experiments with very simple static imaging experiments. We used optical fluorescence microscopy. So we excited at 450 nanometer. So we just hit the center of the absorption of this molecule. If you remember a few slides back, this light comes here from the left, is reflected up through an objective. We defocus the light actually to a large area on the sample plane, the so-called wide field imaging, where we see and illuminate an area of about 70 micrometer. Everything that is that fluoresces in this 70 micron area will be collected by the same objective, and then the fluorescence goes down and is imaged onto a CCD camera. So this allows us to identify where we have nanofibers on our sample. Then we switch to confocal mode. That means we take out this wide field lens from our path and we focus down on a diffraction limited spot of about 200, 250 nanometer in size. And at the same time, we move the sample so that we excite the fibers only at one end, very locally. And then we want to see, because remember, the field of view of the microscope object of the microscope is still that we can observe all fluorescence that comes from this 70 micron diameter here. So not only from this local excitation spot. So we excite locally, and then we see whether fluorescence appears from other positions along the fiber than the excitation spot. So in reality, it looks like this. Here is a wide field image of one of these samples. And what you see here are several elongated fluorescence structures. And I want to focus on this lengthy structure here in the center, in this orange box, which has a length of about 4 micrometer. And now, as said, we switch to confocal excitation and excite only locally at the bottom left position of this nanofiber and collect fluorescence. And this is what we see. So we excite only in at this position of the blue spot. This is roughly our size of the excitation spot, but we see fluorescence along the direction where the nanofiber actually is. And that means, because we're talking about a structure which has molecular diameter, a few nanometer diameter only, it cannot be a wave guiding of light. This must be coming from transport of energy, which is then released at different positions as the energy is transported along the nanofiber. And we didn't do that only on one structure, of course. We did that a bit more systematically, did a bit statistics. So wide field imaging, this wide field illumination on the left allows us to measure the physical length of the fibers, because we illuminate everything. And if we illuminate everything that can fluoresce will fluoresce, and then we can measure how long the fibers are. And in the histogram down here, we see that these lengths are about 3 micrometer. If we do the same game for confocal illumination at the fiber ends, we measure the distance from the excitation spot to the end of the PL signal, and we see again that this length, which we then call the transport length, is around 3 micrometer. So the same maxima. And if you overlay these two histograms, we see that there is not much difference. That means for all fibers that we investigated, it's essentially the limiting factor is the length of the fibers. It's not that energy transport would not be efficient in that systems and not be long range. It's really that we are limited by the length of the fibers. Now, how can we understand that we have this long range energy transport? Now, here a bit the sketchy explanation. And this relies on the emission profile. If you go under confocal excitation, point-like excitation, if you extract the intensity, the fluorescence intensity along the axis of the nanofiber, along this orange arrow here. And you see we have a huge signal at the excitation spot, but there are also two other positions roughly in the center and towards the end of a nanofiber. So that would translate into our sketch here on the left. So we have our pi-stacked H-aggregated core of the nanofibers. Then the green one is, if you remember, these are the hydrogen bonding amide groups that keep all in shape, in this perfect shape. And just keep in mind that this represents now 4 micron, and the pi stacking distance of molecules is 0.3 nanometer roughly. So we're talking about more than 10,000 molecules here. And what we see is the fluorescent periphery. So remember, we have a large signal at the excitation spot in the middle and at the end of the fiber. So that's represented here by these 3 arrows symbolizing the emission, where it comes from the periphery. Now, what happens if we excite this nanostructure? We excite it at the bottom left, these blue arrows here. And again, what we do excite is both the periphery and the core of these nanofibers. So the fact that we observe a strong fluorescent signal at the position of the excitation spot, so here, this is not really surprising because we directly excite in part the periphery. But we certainly also excite in the core, and then we excite a delocalized exciton of an H aggregate. But of course, in a real system, the delocalization illustrated here by these ellipses, that should be the delocalization of exciton, is not along 10,000 molecules. In a real system, you always have defects. And at these defects, if this delocalization reaches a defect, you have 2 options. So the first one would be energy transfer to the periphery, and then you get emission. Or the exciton can hop across the defect into a neighboring domain where it again starts delocalizing, and so on until you reach the end of the nanofiber, which is the largest defect you can have, and where you can again have energy transport to the periphery and emission. So what we have is that delocalized excitons move incoherently along the nanofiber. And since a delocalized exciton means that the molecules' excited state wave functions are coherently coupled, we term that coherent transport. So we have a coherent-incoherent transport mechanism. So that's our hypothesis. The point or the problem in these measurements and in this hypothesis that we have is that this relies on measurement of incoherent fluorescence from the periphery. So we in fact do not know what exactly happens in the core of these nanofibers because we only see the emitting periphery. So we talked to our favorite chemists again and asked them whether there is any chance to get rid of this bithiophene, this naphthalimide bithiophene emissive periphery here, and to replace that by something that is not emitting. So we really want to see the H-aggregated core. And chemists came up with the molecule here on the right. So we have exactly the same, the core, the CBT core is exactly the same. So we have again an aromatic core, which, well, has attractive van der Waals interactions. If they start interacting and stacking upon self-assembly, we have the amide groups here, 3 of them. So these pi stacking is enforced by 3 hydrogen bonds to neighboring molecules. So all the same except the periphery, which is now a simple phenyl ring with 3 groups. These are just long aliphatic chains shown here. If there are any chemists among you, you will see that here we have a S-chiral group here, and that is why we call this molecule S. This is not SCBT. Now, if you do this change in molecule, it took us a few years to come back to really get nanofibers out of this new molecule with a non-emissive periphery. But we realized at the same time that this allows us to really tune the excited state energy landscape if we use different preparation conditions. So in the first condition that we did, we used SCBT in dodecane, and it self-assembles into nanofibrillar structures shown here in the AFM. Here you see these nanofibrillar structures. And just note the height scale here, we're talking about a few nanometers only. So that is again the molecular diameter. So we have molecular sized nanofibers here with a length of several micron. If you look at transmission electron microscopy, I hope you can see it. So here you see that these structures, these fibers lie just parallel next to each other in certain areas, here and up here, up here. For analysis, we first performed a fast Fourier transform of that image, and we see 4 signatures here, here, this one, and this one. If you translate that in a distance, we end up at 3.4 nanometers. And if you compare that to our molecule, this is the diameter of a molecule. So it's really that we have nanofibers with a diameter of our molecules. We can also do electron diffraction on these films of these nanostructures on substrates. And that's shown here below. And here you see these very faint ring. And if you translate that into distance, we end up at 0.33 nanometer, a characteristic pi stacking distance. So we really see that the core of the molecule has a distance of 0.33 nanometer. So the structure of our nanofibers is exactly what I've drawn here. On the side, we have this perfect H aggregates of the CBT core. If we use for preparation a different solvent, in this case anisole, we get different structures. So the AFM image is shown here. There's several micrometer in length here, the structure. But now if you look at the height, we're talking about 100 nanometer now. A TEM image is shown here. Watch out, this is a scale bar is 100 nanometer. So what you see here is a structure with a width of about 200 nanometer, in line with the 100 nanometer of AFM. So there seems to be a larger hierarchical structure. And if we do diffraction on this TEM data that we have here, we get 2 signals. One is here, these point-like structures here and here, perpendicular to the axis of our feature, of our structure that we have. That corresponds to 3.2 nanometer. And at the same time, we get a diffraction signal in the direction of the nanostructure with 0.33 nanometers. That means we have, again, 2 distances. The 0.33 is a pi stacking distance between the CBT cores here, and the 3.2 nanometer is the distance between fibrillar structures in a larger bundle. So we have a hierarchical structure here. And remember, this 3.2 nanometer is slightly smaller than the 3.4 nanometer of the diameter of the molecule. So what happens is that these aliphatic side groups here within the bundle, they start interacting, they start interdigitating. And overall, the size of this bundle, the diameter of this bundle is about 100 to 200 nanometers. Now, looking into energy transport in that system now allows us to really look at what is happening in the core, because that's the only thing that is optically accessible now. Well, first, this is just a summary. Just we get once isolated nanofibers. And on the other side, different preparation, we get bundles of nanofibers. Yeah, that is for if there are some experts among you, I mentioned that we have chiral side groups in these aliphatic chains of these molecules. So you expect a chiral arrangement along the nanofiber axis as indicated here in both fibers and bundles of fibers. And circular dichroism indeed shows this characteristic cotton effect. So verifying that we have indeed chiral structures in both systems. Now, if we do plain optical spectroscopy in the system, we start with the molecule itself, so not in a nanofiber or in a bundle, so just the isolated molecule in solution that would be in THF. And yeah, the wavelength axis is missing. It will come later, unfortunately, but this is around 450 nanometer. But the absorption in blue and the fluorescence in red, they have the very characteristic shape of an aromatic molecule. There's nothing special about it. And this is just absorption by the core of our molecule and the emission. So a transition from ground to excited state. If we go to nanofibers in dodecane, isolated nanofibers, we see that the spectra dramatically change. And what is important is, in fact, that the electronic transition, let's start on this side. So we have the formation of an exciton band here, this gray area. And if you remember the beginning of my talk, only the top state has oscillator strength and an H aggregate. So you see the absorption into the top state of this exciton band. Then you have very quick relaxation to the bottom state. So you get a large Stokes shift between absorption and emission. And this bottom state starts emitting. But of course, it's a subradiant state. So a low signal, a low fluorescence signal with a large Stokes shift. And this is exactly what we do see. Here, the electronic transition is strongly suppressed. Yeah, so this is the normalized data. But we know that the fluorescence signal is very, very weak from this system. The solid line here for absorption and the dashed line for the emission spectrum, these are numerical simulations based on a model put forward by Frank Spano. And then we know that the electronic coupling between neighboring molecules in a nanofiber is around 700 wave number. This is a reasonably strong electronic interaction between transition dipole moments. And it clearly indicates that delocalized excitons are present in these nanofibers. Now, if you look at the other example, that would be the bundles of nanofibers in anisole at the bottom here. So the absorption spectrum of these bundles, they look very similar to the fibers if you compare these two spectra. So again, what's happening is we form an H aggregate with this exciton state and the transition into the top state of this exciton band. But the emission is now very peculiar, because if you compare the emission of these bundles with the emission of the isolated monomer, it looks very similar. It's just shifted by about 70 nanometers to the red, to longer wavelengths. So that means that the emission, and the emission is indeed a bit stronger than for nanofibers. So that means the emission from the bottom of the exciton state is not as suppressed anymore as it should be for a perfect H aggregate. And we know, in fact, from this modeling, again, we modeled the absorption, this dash, the solid line, and the emission, this dashed line here. We modeled that with this theory put forward by Frank Spano. And this model also allows you to get out the exciton landscape. And now this exciton landscape, you have to be a bit careful, is now plotted not in terms of excitons, but in terms of the position n of the monomer if you go along, if you go along a single nanofiber. So how do the individual excited states look like, and how do they develop as function of position if you go along the state, along the nanofiber, sorry. And what we found, and how we could only simulate the spectra, especially this broad and redshifted spectrum in emission, is that the energy difference between neighboring CBT molecules is not very large. So they are very, very similar. It's a very smooth energy landscape here. In contrast, for anisole, for the bundles of nanofibers, we see the different colors, green, gray, and orange, they indicate just 3 different realizations for 3 different nanofibers of this disorder. For the bundles of nanofibers, we find that if you go along a nanofiber in a bundle, we see that there are huge differences in transition energies for different monomers. That means the excited state energy landscape is very rough. So you hardly find 2 molecules with very similar neighboring transition energies. And that, of course, has a consequence for the delocalization of the exciton. If you have the smooth energy landscape as in the single fibers, you have a large delocalization of the exciton. You have a limited degree of disorder. But if you have this large degree of disorder as in the bundles, well, delocalization is relatively small, 1 or 2 over 1 or 2 molecules only. And that, of course, explains why the shape of the emission of the bundles of nanofibers and of the isolated monomers is very similar. And that means what happens is, if you have isolated fibers and bundles of nanofibers, the only difference is what I explained in the beginning, that the side chains, they start interdigitating and interacting with each other in the bundle, and they introduce disorder. So in this sense, bundling introduces disorder along each nanofiber, and that localizes excitons. And if you remember back the first time of my talk with these, with the other molecule, we said that we have coherent and incoherent transport. That means delocalized excitons should be favorable for transport. So what we expect would be that single nanofibers are much better energy transporting systems than these bundles of nanofibers. So how do we measure that? So we measure that actually in space and time. And we use the so-called beam scanning, detection beam scanning method. So we have a wide field image here. This here is our nanofiber, a single nanofiber. It's very weak, so it's very noisy signal, but it's very weak. It's an H aggregate. And we again excite locally at the center with a diffraction limited spot. And then what we do is we move the detection here, this part here, we move it so that we collect emission from different positions along the nanofiber, up and down relative to the excitation spot, so we can directly monitor how the energy propagates up and down in these nanofibers. And so what we get from this nanofiber is shown here. So the x-axis is the position along the nanofiber, and remember at 0 here we excite the molecule. And then here on the x-axis we have time in 5 nanoseconds, up to 5 nanoseconds, and what you see is that the emission profile really starts broadening with time within a few nanoseconds. That means the energy really propagates within a few nanoseconds along this nanostructure. And we can analyze that here on the right side. This is just time, again, the same time axis as here on the left. And the y-axis is the width of this profile, just the width with these two white lines. And you see that this broadening occurs indeed in time. And from these data, I don't want to go into detail, but from these data, we can determine a diffusion constant for excitons of 1 square centimeter per second, which is actually for H aggregates still the current world record. Now, if we do the same experiment for bundles of nanofibers here, so here's a wide field image. This is the bundle, what you see, again, few nanometers. And again, we do the detection beam scanning. Again, here is the part where we excite. And you see on the white line, as function of time, the emission does not spread or barely spreads, right? There's only a small effect visible. This is also when you plot the widths, so the widths, the dimension between the two horizontal white lines here, you see that's only a minor effect in broadening. If you compare that to our single nanofibers on top here, all axes are the same, and also these solid line here is the same. So you really see that transport is strongly limited in the bundles. And the diffusion constant we get out is about 0.05 square centimeter per second. So that really means the delocalization of exciton assists, it massively enhances transport in these H-type nanostructures that we have. Now I want to skip that part, given time. And this is the part where I want to show some new data, which are still unpublished. Yep, thanks. So this is where I want to come back in the last few minutes to this example that I introduced in the beginning, where I said that in these light harvesting complexes, nature generated an energy funnel, so an energy gradient, excited state energy gradient from these peripheral light harvesting complexes into the reaction center, using only a single species of molecules, bacteriochlorophyll. And nature actually does that because what you see here is that the red and the blue chlorophylls are differently oriented with respect to each other. And from my introductory slides, it should be clear that you have different exciton states that are optically active. So this is part of the energy funnel. Another factor that contributes to this energy funnel is these interactions between the pigments and the proteins. Now, the problem is that we certainly do not want to copy nature, right? That would not be feasible in any way. The system is way too complex to use it and to build it. So we were looking for a system where we can, again, use a single species of molecule to create an energy gradient.